Clustering is about uncovering natural groupings in data. It has many applications, from grouping customers by purchasing behavior to clustering houses with similar characteristics. At its core, it follows a very simple rule: points in the same cluster should be similar. But what does that really mean? This cluster might seem reasonable, but look at how far apart these 2 points are at its edges. Should such distant points belong to the same cluster? Maybe what makes a good cluster isn't how close its 2 farthest points are, especially if they're connected by a chain of points that are each close to one another. Or, like k-means, maybe what makes a good cluster is how far points are to the center of the cluster. But even if we agree on what criteria makes a good cluster, there's still another question. How far is too far? Where do we draw the line? At this scale, we might see 5 clusters, each with points that seem similar, but if we zoom in, we see more intricate structure appear, and points that had seemed similar might now be clustered separately. Clustering is a challenging task because it's such an ambiguous one. It has many valid solutions depending on perspective. Hierarchical clustering embraces ambiguity, not as a problem to solve, but as something to explore. This is how I think about hierarchical clustering. Ambiguity in clustering boils down to how much of a difference we're willing to tolerate between points in the same cluster. Some may have a high tolerance, and thus meeting the criteria for belonging to the same cluster is less restrictive. With that perspective, many points seem similar and very few very large clusters are formed. Others may have a low tolerance, and thus meeting the criteria for belonging to the same cluster is more restrictive. Points need to be very close in order to belong to the same cluster. With that perspective, very many very small clusters are formed. Hierarchical clustering explores how varying the tolerance level changes the clusterings created. Intuitively, when tolerance is small, only points that are very close to each other, like these 2 points for example, would meet the criteria for being in the same cluster. As we move tolerance up, points that are a little farther apart now meet the criteria and can be clustered together. Eventually, points get absorbed into clusters that are close by, or 2 clusters that are close together get merged into one. But what does it mean for 2 clusters to be close? Is it enough for 2 points to be close? Should all points be close? Should the centroids of the clusters be close? In hierarchical clustering, distance between clusters is defined through a linkage function. More subtly though, the linkage function defines the criteria that makes a good cluster. Consider these 3 clusters, for example. Under what criteria would we merge blue and yellow, and under what criteria would we merge yellow and purple? Single linkage is defined by the closest pair of points between 2 clusters. It creates clusters where points are close to their neighbors, creating a chain of close points. Under this criteria, we would merge blue and yellow as they more naturally chained together. Complete linkage is defined by the farthest pair of points between 2 clusters. If we want more compact clusters where points are close to all points in the cluster, 2 clusters should be merged if their 2 farthest points are close, thus ensuring all points are close to one another in the cluster. Under this criteria, we would merge yellow and purple, as it would create a less spread out cluster. If we want clusters where points are close to the center, there are a few options. Centroid linkage is defined by the distance between the centroids of the clusters. Average linkage is defined by the average distance between a point in one cluster and a point in the other. So for each point in the blue cluster, we get its distance to all points in the yellow cluster, taking the average of all these distances. And we compare that with the average distance between points in the yellow cluster to points in the purple cluster. Ward's linkage is defined by the amount of spread around the mean resulting from merging 2 clusters. For example, each of the following clusters has a small spread, but if we were to merge them together, the spread of that cluster would be very large compared to the sum of the 2 smaller spreads. By contrast, because these 2 clusters are closer, the resulting spread would be small and not much different from the sum of the 2 small spreads. In that sense, Ward's linkage is the hierarchical counterpart to k-means. Each of these linkage functions brings to light different structure in our data, allowing us to make sense of clustering's ambiguity. Hierarchical clustering functions as follows. Start with all points in their own clusters. Compute the distance between all pairs of clusters using the linkage function. Merge the two closest clusters and record the distance at which the merge occurred in a dendrogram. Repeat steps 2 and 3 until all points are in the same cluster. This approach is known as agglomerative clustering. There's another top-down approach called divisive clustering where all points start in one cluster and are repeatedly split. But unlike merging, where intuitively the best next step is to merge the two closest clusters, it's not clear how to best split a cluster. So the divisive approach is less commonly used. Let's walk through the agglomerative clustering algorithm using the single linkage function on the following dataset. We have A at the origin, B at 1, 1, C at 3, 0, and D at 0, -2. Let's fill out the distance matrix of all pairwise distances. Now, we merge the two closest points into a cluster. That's A and B at a distance of square root of 2. We record in the dendrogram that A and B would merge into a cluster at a distance of square root of 2. Now, we update the distance matrix. Notice that the distance between C and D does not need to be updated. The distance between cluster AB and point C, according to single linkage, is the smallest pairwise distance. So we compare the distance between A and C to the distance between B and C. The distance between B and C is smallest, so it defines the distance between cluster AB and point C. Similarly, with the distance between AB and D, it's defined by the distance between A and D since this is the smaller of the two distances. Now that the distance matrix is updated, we find and merge the two closest clusters, which are AB and D. We record in the dendrogram that AB and D would merge into a single cluster at a distance of 2. And we update the distance matrix, and finally merge ABD with C, and record that in the dendrogram. The dendrogram is what reveals the structure of our data under this linkage function. The distance on the y-axis represents the distance we're willing to tolerate for points in the same cluster. In the case of single linkage, it describes the distance between neighboring points in the same cluster. For example, let's say we draw the line at a distance of 2.1, meaning we cannot tolerate a distance of more than 2.1 between a point and its neighbor in a given cluster. This creates the following clustering: ABD in one cluster and C alone. Single linkage guarantees that a point in a cluster is at most 2.1 distance from the nearest point in the same cluster. Indeed, the distance between A and B is square root of 2, and the distance between A and D is 2. Let's go through the same example, now using complete linkage as a contrast. The initial distance matrix is the same, and thus the first step is the same since we always merge the two closest clusters. Let's update the distance matrix. The distance between cluster AB and point C according to complete linkage is the largest pairwise distance. So we compare the distance between A and C to the distance between B and C. The distance between A and C is largest, so it defines the distance between cluster AB and point C. Similarly, with the distance between AB and D, it's defined by the distance between B and D, since this is the largest of the two distances. Now that the distance matrix is updated, we find and merge the two closest clusters, which are AB and C. We record in the dendrogram that AB and C would merge at a distance of 3. Finally, we update the distance matrix, and we merge ABC with D. The y-axis now represents the farthest distance between points in the same cluster. Cutting the dendrogram at a distance of 3.1 means no two points in a cluster should have a distance greater than 3.1. This is only possible if ABC is in one cluster and D is alone. I know what you're thinking. How do I know where to draw the line? Great question. The answer lies in the shape of the dendrogram. Consider this more realistic dataset, where we have two distinct clusters. The dendrogram clearly shows that two sets of points get merged at relatively similar distance thresholds. Then there's a big gap before these two sets of points would merge into a single cluster. Drawing the line somewhere in that range generates the two clusters we intuitively hope to find. But sometimes there are many options. These clusters might be part of a bigger group, and these two might be part of an even bigger group. Similar to phylogenetic trees, where you can create very broad groupings like kingdoms and very specific groupings like species, there may be many valid clusterings at different scales. But the choice of linkage drastically changes the dendrogram and how well it conveys structure. For example, notice what happens if we add just a few points in between the two clusters. Now, it's not clear from the dendrogram that there are two clusters and a bridge between them. Because single linkage builds clusters where a point and its neighbor should be close, it finds many points that fit that criteria in the bridge between the two clusters. So from its perspective, these are indistinguishable from how close points are in each of the two obvious clusters, and finds one connected structure instead of three separate parts. Complete linkage attempts to counteract this by looking at the other extreme, the maximum distance. Let's rebuild the dendrogram using complete linkage instead. Notice how the structure becomes much more apparent because complete linkage requires all points in a cluster to be close. It much prefers to create three compact clusters compared to one elongated cluster. But no linkage is perfect, and an asset in one situation can turn into a limitation in another. If we elongate one of the two clusters, complete linkage struggles to find that elongated cluster, preferring to split it up into two. Even if we remove the bridge, complete linkage prefers to split the elongated cluster into two. Now, single linkage shines, finding two clusters more naturally. Each linkage function reveals or obscures interesting structure in our data, but some structure remains obscure no matter the linkage function. Consider the following clustering. We could argue that there are three clusters, two are the obvious dense regions, and one is a set of lesser dense points surrounding them. Yet, no single cut can produce this clustering. The dense regions merge at very low tolerance, and the surrounding points only merge at a much higher tolerance, and they cannot be merged together without also merging them with the dense region. So any single cut either captures the dense clusters and splits the rest, or groups everything together and we lose the finer structure. So maybe having a single tolerance with which to define all clusters is too restrictive. I hope this helped you better understand hierarchical clustering. It's a powerful tool because it embraces ambiguity not as a problem to solve, but as a property of the data itself to be revealed and studied. Like, subscribe, and help me spread this knowledge around if you found it helpful.